everybody. My name is Annie Voss, and I worked in the Julkowska lab this summer, and I was studying the molecular mechanics underlying tepary bean responses to drought stress. You might be wondering, what is the tepary bean? Well, the tepary bean is a legume that's native to North America. It's native to the desert, so it's naturally drought stress resistant, and it's also related to the common bean. The common bean includes all the beans you eat every day. The black bean, the pinto bean, the navy bean, the green bean. And that kind of bean, it does not grow well with drought stress. It's having a lot of problems agriculturally with climate change. And so we want to understand, can we breed the common bean and the tepary bean together? And the answer is yes, but we don't know what the genes are actually in tepary bean that's causing it to be resilient to drought stress. So that's what I was looking at this summer. We did 3 different experiments that are kind of looking at this problem. So the first thing that we did was actually just grow the tepary bean. We grew a couple different accessions that we thought, some might be susceptible, some might be tolerant, and we grew them in this big plant conveyor belt called the phenotyping rig here at BTI. And what we found actually was that these plants, they kind of all grew the same, which was not what we were expecting. But at least we know how they grow. And that allows us to move on to the next part of our experiment, which is looking at the specific genes. And someone else in my lab had been able to identify homologous genes in Arabidopsis, which means genes that are in both of the plants, and that they think are affecting drought stress resilience. And so we did an experiment where we grew Arabidopsis mutants of these genes on plates with mannitol, which induces osmotic stress, which is pretty similar to drought stress. It's about as close as you can get in this experiment. And what we found was there was one gene in particular called AKR4C9 that grew way, way, way better than all the rest of them. So we were like, okay, we have this interesting gene. We have these plants that we know how they grow. Why don't we take this plant and this gene and knock out the gene and see what's happening with drought stress? But the problem with that is that the best way to do this is virus-induced gene silencing. And virus-induced gene silencing has never been done before in the tepary bean. So we actually had to figure out, can you do this at all? And so what we did was take a different experiment that had done it in a plant called cowpea, and we just completely replicated the whole experiment. And in this experiment, they were looking at the PDS gene. And when you knock out the PDS gene, what you get is a different color in your leaf. And you can see here, that's what we got. We got this sort of speckled mottled color thing. And that's basically the gene being turned off in the plant. So we can successfully say, yes, we know how to do virus-induced gene silencing in tepary bean. And so now what we're doing is an ongoing experiment where we're taking the genes that we identified over here in these plants, and we're knocking them out, and we're using virus-induced gene silencing to look and see how these plants are growing. And we've already done the inoculation process, and now we're looking at what's actually going on in the plants in the phenotyping rig. So we're really excited to see what comes next.
All right, so you did a great job. Do you know how the AKR gene might be helping or affecting the osmotic response in the plants?
Yeah, so we're actually not entirely sure, and that's one of the things we hope to understand. So in these plants, we've actually knocked out the gene. So something about AKR4C9 being gone actually causes the plant to grow a lot better with drought stress. And this is a root system analysis, so maybe it does have something to do with the roots, but it could be actually something totally different, and that's something we'd like to find out. So
you did a great job. Thank you.
Thank you.